Hi, I'm Dr. Villar, and welcome to this second presentation on mechanisms of resistance to antiparasitic drugs. On the first presentation, we saw that the foundation of a good control of parasites started by understanding their biology. We did a quick overview of the main parasites of concern in ruminants in tropical countries such as Colombia. And on this second presentation, I will start by saying that when we have tried to eradicate parasites, with very few exceptions, history has shown us that we're likely to fail because they tend to become resistant. So really the idea or goal should be to control them to the extent that they don't cause production losses or sickness in our animals. And really to achieve this, there is no ABC approach, such as deworming every so often like we've been doing and continue to do to this day. And we need to understand that parasites have coexisted with their host for thousands, if not millions of years. They're part of a system. They're there for a reason, and it's really not in their best interest to kill their host. So in this respect, a few parasites we could say that are not a problem, and in fact they may be beneficial to stimulate the immune system and build up a protective response on that animal. Here is an outline of what's covered in this presentation. We will start by briefly mentioning some of the vast literature that we can now find on resistance. We'll then mention a method to detect resistance. We'll then try to explain why and how resistance makes its appearance after we use a dewormer for an extended period of time. We will then integrate this idea with the concept of preserving the so-called population in refugia, and we'll finally revise some types of mechanisms of resistance that take place at the molecular level, and give some practical examples of this using the cattle fever tick as a model of a parasite. Briefly, some titles in scientific papers. You will find many papers that basically examine the problem of resistance, which is now worldwide reported for almost every type of parasite of economic importance. If you look at some of the titles, An Inconvenient Truth, Global Warming and Anthelmintic Resistance. The second one is an update of the situation of anthelmintic resistance in Latin and South America. And the third one is a review of the situation with the cattle tick, Rhipicephalus microplus in Brazil. This parasite is a real menace to raising European cattle breeds, and those same problems that they have in Brazil also extend to countries like Colombia. In fact, I'll show a few personal examples that we have been working on the next video. So really there is no longer any doubts that the mindset of relying totally on drugs to control parasites has to change. And in fact, you'll find many papers like the ones on this other slide, which are alternative approaches that include changes in pasture management and husbandry practices, which tend to be more sustainable if we want to achieve long-term productive livestock systems. Here are some of the papers. I will not really go into more detail now and just simply invite you to watch the video on integrated management practices. However, I would like to say that apart from scientific papers, we can also find many technical bulletins or manuals like the ones we see here. The first one kind of selected a buzzword, COWS, which is really an acronym for control of worms sustainably. Really, in light of the resistance problems, these manuals are intended to integrate the use of dewormers with other management tools that tend to be more rational to simply relying on drugs all the time. Now, before I move on to how and why resistance makes its appearance on a farm, I wanted to show a study to illustrate how resistance against roundworms is typically identified. This is a study that was done in 59 clinics in the US from 19 different states. They selected 20 animals from each cattle farm, and they collected fecal samples from each one of those animals on day 0, and then 14 days after they were given a dewormer. On each of those fecal samples, a technique of sugar fecal flotation was done to measure the number of eggs per gram of feces. And if we look at the results of this study, they measured the efficacy of the dewormer based on the reduction in fecal egg counts. That's why they call that test fecal egg count reduction test. Now on the upper table we have the results for the injectable microcyclic lactones, and on the lower one the same for the Poro-On formulations. When we compare the number of eggs before and after the treatment, if there was no resistance whatsoever, that treatment should have killed all the parasites, and obviously there would be 0 eggs on the second time around that they did the collection, and the efficacy would be 100%. However, if we look at the bottom of those tables, we notice that the average efficacy is 72% for the injectables and only 66% for the Poro-On preparations. In other words, there is already much resistance and it's only going to get worse as they continue using this type of microcyclic lactones. Now on that same study, we have another table that they combine microcyclic lactones with Safeguard and Panacur. These are benzimidazoles, a different family of dewormers, and we see that the efficacy was 100%. So basically the use of dewormers that work differently by different mechanisms overcame the problem of resistance, at least for the time being, because as we will see in a minute, it's just a matter of time that resistance will show up, and it kind of depends on how frequent we use the same dewormer over and over again. Now before we go into the mechanisms of resistance, let's see how we could describe what happens when we apply a dewormer on a group of animals. Imagine that you treat an entire flock of sheep. This curve shows the dose of the dewormer in milligrams per kilogram of body weight that would be necessary to kill different percents of that parasite population in that flock. Ideally, you want to use a dose that will kill 100% of the parasites in your flock, obviously. But as you can see here, the dose required to kill different individuals in that population will vary depending on how susceptible each one of those parasites happens to be for that dewormer. Having said this, how susceptible they are could be simplified, just to make this easier to comprehend, because I know it's not that simple, by how many target proteins are susceptible for that particular drug chemical. The very susceptible individuals will have 100% of those target proteins from both alleles as susceptible. But as we can see, as we move to the right of the curve, we need more dose to kill those parasites, because we start seeing that one of the alleles from one of the parents starts coding for a resistant protein to which that chemical is no longer a target, obviously. At the farther end of the curve, we can still kill all the parasites because they still have a majority of susceptible proteins, but as you can see, the number of the resistant ones have increased because that one resistant allele is being expressed to a greater extent. Now if you notice, there are two circles in the graph. There will always be some individuals in that flock that already have both alleles that are resistant, and so no matter how much we increase the dose, it will be impossible to kill them. This is the reason why we could say that the resistant genes are already present before we even start using an antiparasitic drug for the first time. And obviously these two resistant parasites that will not be killed by the treatment will be the ones that will be mating if all the other ones under the curve have disappeared. And obviously these will be passing on their genes into the next generation that will also be resistant. Now if you understood that previous concept, the one on this slide will be very simple to comprehend. The question that could be posed is where does resistance makes its appearance earlier? When we use drugs like fenbendazole, which is very rapidly clear from the bloodstream, or when we use an endectocide like the microcyclic lactones that, as you can see, and depending also on the formulation and dosage, can stay in the body for up to 3 months at least on this illustration here. Now, in this graph, the duration of action could be expressed by the time at which the concentrations in plasma are above the lethal dose, and is clearly much longer for the endectocides than for fenbendazole. However, if we really wanted to select for one that will not favor the development of resistance, we would rather have one with a concentration curve like fenbendazole rather than one with a curve like the endectocides. What happens in the part of the red line curve that is below the lethal dose, which is shown in as a blue line, is that we're going to kill the most susceptible worms in the population. That is those that have both alleles being susceptible, and those that will be heterogeneous with one allele of each kind will simply survive, and that will result in an increase in the overall resistant gene in the next generation of worms. In conclusion, if we want to select a dewormer from the standpoint of slowing resistance, we need one that gets in the body, acts quickly, killing as many of the worms as possible, and basically is rapidly eliminated from the body in just a matter of 1 or 2 days. Now, the final concept to wrap up all these ideas and visualize how everything is happening in any one farm is what's known as the population in refugia, which is basically all the different parasites in a farm, including all the different life stages that are not being exposed to an antiparasitic drug. As you can see here, the population of worms that are not being treated, which includes all those larvae and eggs that are on the pasture, they tend to be much greater than the one present in the animals that we treat. And when we use a dewormer for the first time, that population is almost all susceptible, which is the one represented here with a green color. And as you can see, there may be a few very little resistant ones that are shown in red. So when we treat our animals, hopefully selecting those that have the most parasites, that usually tend to be the young individuals that have little or no acquired immunity, we will pretty much clean them from most, if not all, the parasites, except for maybe a few resistant ones that are at the beginning are almost non-existent, as I say. And as soon as the effect of that drug wears off, a new generation of parasites that are on that pasture will infect the animal again. So because this new generation is mostly susceptible, the drug will continue to work when we use it again. But as you can deduce, over time the number of resistant worms will tend to increase because they have no competition from susceptible ones that have been gradually eliminated. So in the end, we end up with a population of only resistant parasites. Now, the take-home message from this is that we should not remove all the population in refugia, because that will speed up the appearance of resistance. And in fact, and as we will see on the next video, current practices emphasize on the importance of preserving this population in refugia, basically to preserve the efficacy of the antiparasitic drugs. Now, at this point, let's say what are some of the molecular mechanisms by which parasites are resistant to drugs. And I'm going to use the cattle tick, whose scientific name is Rhipicephalus microplus, as an example to illustrate this point, but the underlying mechanisms will likely apply to any other parasite. The first one, and that usually confers the most resistance, is when the drug is no longer able to bind and interact with its target site. I will show an example of this type on a diagram on the next slide, in which pyrethroids that usually bind voltage-dependent sodium channels are no longer able to do so. And obviously, if this interaction was creating an overstimulation of all the nerve impulses in that parasite, ultimately killing him, now it will cause nothing on that parasite. Now, what is usually interesting is that there is what's called a cross-reaction or of resistance between drugs that work at the same site. And we have noticed this for most pyrethroids, which are a large family of drugs. In other words, if permethrin does not work, it's unlikely that another related pyrethroid like cypermethrin or flumethrin or deltamethrin will also work. This is obviously a major drawback in that it limits the number of options that we have available to control this parasite. However, if we look at the next group of acrocytes, which are the organophosphorus and carbamate compounds, even if they all work by inhibiting the enzyme acetylcholinesterase, we know that there are at least 3 different types in the tick Rhipicephalus, and that explains why they don't show as much cross reaction as you could expect initially. On the next video, I will show some results of our studies in which we demonstrate that an almost complete lack of efficacy against chlorpyrifos in one strain of tick from one particular farm was very susceptible against another organophosphorus called ethion. Another important mechanism of resistance is to increase the metabolism or detoxification by inducing enzymes that break down or eliminate the drug from inside the parasite. Some of the different enzymes, which in turn have many isoforms, are listed here. We have esterases, cytochromes, glutathione transferases. You will see that many times insecticides are combined with other compounds like piperonyl butoxide, which is basically a synergist that blocks some of these enzymes, in this case the cytochromes P450s. And this is a way of neutralizing these mechanisms of resistance in the parasite, if you like. And finally, the last type of mechanism, which is one that I don't quite comprehend very well, but it seems to avoid the penetration of the antiparasitic drug to its site of action. This type of mechanism is somewhat similar to what happens with ivermectin in certain dog breeds like the collies. This breed of dog cannot be given a normal dose of ivermectin because the protein transporters that are present in the blood-brain barrier will not recognize the drug, and it will allow it to penetrate into the central nervous system of the dog, and this could kill him. Now, there are some ticks that are resistant to ivermectin, and they basically have overexpression of these transporters, which basically prevent ivermectin from reaching its target site in the tick, which are the GABA receptors. On these final 2 slides, we have some examples of these mechanisms of resistance in ticks, and it's interesting to see that we can find several of these mechanisms at the same time in any one resistant population of ticks. On this slide, we can see a diagram of the voltage gated sodium channel, which is a very big membrane protein that is present in excitable cells like the neurons of the parasite and our neurons for that matter. These channels are responsible for the rising phase of action potentials that take place in nerve impulses. And if you notice the protein consists of beta subunits, and there is a large alpha subunit in the center or core of that channel, which in turn has 4 domains, and each one of those domains contains 6 transmembrane segments. Now, the reason I mention all this is because there have been 2 single mutations discovered in resistant ticks against pyrethroids. One is present in domain 3 and the other in domain 2, which make the channel insensitive to pyrethroids. And not only that, but when that mutation is in domain 3, the degree of resistance is enormous, requiring more than 1,000-fold the concentration of a pyrethroid to kill that tick compared with a susceptible one. However, when that mutation was found in domain 2, the degree of resistance was only 100 to 400 fold the concentration that would be necessary to kill that tick if he had been susceptible. Consequently, the phenotypic effect of each mutation differs quite a bit in terms of the degree of resistance, and you can read more about this in the reference that I'm providing here at the bottom of this slide. Now, obviously, when we look at the entire population of ticks in any one farm, the degree of resistance will depend on how many individuals you have that are homozygous, susceptible, heterozygous, that is with one allele, both susceptible and resistant, and homozygous for both resistant alleles. I would like you to look at the top table where it says resistance ratio for 4 different strains of ticks from Mexico. The higher the resistance ratio simply means that we need to expose that strain to a concentration of whatever number you see there above the one needed to kill it if it was a susceptible tick. And as you can see, the most resistant one would be the Corralis, with the San Felipe being intermediate between the Coatzacoalcos and the Corralis. So if we look at the table below, you will notice that the most resistant strain, which is the Corralis, has 97% of the individuals carrying the G mutation that confers a very high level of resistance, and only 3% are heterozygous. Now, if we look at the San Felipe, which is between the other two in terms of resistance, there are more heterozygous individuals, 29%, and 71% are all resistant. And if we look at the least resistant strains of the three, the Coatzacoalcos, almost all of them carry the homozygous for the susceptible gene. This would basically indicate that the sodium channel is not the mechanism responsible for showing some degree of resistance. And in fact, if we look at the right on that bottom table, they also look at a different mechanism of resistance based on the metabolism or detoxification of the permethrin. And if you notice, they found that 100% of the individuals in this strain carry a gene that is overexpressing a mutated esterase that has a 5-fold greater hydrolytic activity to break down the pyrethroid a lot faster. However, if you notice, the degree of resistance that this mechanism conveys is much lower than the mutated sodium channel. So basically, by showing you this type of studies, you can get a feeling of how complex these mechanisms of resistance can be, considering all the possible combinations that can potentially exist in the same parasite. At least now we have these PCR-based assays that can help us detect many of these mutations in just a matter of a few days, and it can give us a good idea of the degree of resistance that a given population might have. Now moving on into the history of the control of the cattle tick, Rhipicephalus microplus. Here we see an old photograph from the USDA showing aspects of that cattle fever tick eradication campaign. This is a good example of a successful program that in just a matter of a few decades managed to completely eradicate the presence of the tick in all the southeastern states of the US and California. And without this tick, the main disease, which is transmitted by the tick, which is babesiosis, also disappeared. And they now estimate savings of up to $3 billion a year from not having this deadly disease. And you may read more on how they make these calculations and all the history on that reference that I provide below. As you can see on the slide here, the operating principles of the fever tick eradication program were strict quarantine, which basically means no movement of animals in and out of infested premises, systematic treatment of cattle with an acrocide. At the time they used these concrete constructions. As you can see there, they look like a trench on the ground, and they were called plunge dips because they would basically immerse all the animal in the water bath with the acrocide. On the next slide I will show you a video clip of a portable dipping vat on wheels, if you like, which is what they use now on the surveillance program. But these concrete bunkers here are kind of a relic of the past now. And the third key to success of the program was pasture vacation, which basically refers to not introducing cattle in any infested pasture until all the free life stages of the tick have died. If you look at the map below, there is a strip of land which is at the border between Texas and Mexico, and which is called a permanent quarantine zone, in which they keep inspectors doing regular visits of every cattle ranch to make sure that deer and cattle that swim across the Rio Grande are not reintroducing ticks from Mexico. And what happens when they find any cow infested with any tick is shown partly on this short video clip. They will put that ranch in quarantine, and they will run every animal through this trailer, which, as you can see, has a dipping vat, and they do this every 15 days for 6 months. And as you can see, this is quite a time-consuming operation, for not saying expensive one, but it pays off. It's if the country is going to remain free from babesiosis. On the short video clip on the right, you can see one of the inspectors refilling up the tank with Coumaphos, which is the organophosphorus acroside, which they use. And unlike other ones, it has proved to be very stable if they keep the pH of the water constant. So all they need to do is replenish the tank from one time to the next, and steer it all up well with a power-driven air pump before they run the animals through the dipping vat. On this other slide, we show 3 methods of control for the cattle tick in Colombia, and each one on its own way makes its own contribution to speed up the problem of resistance by removing the population in refugia that maintains the susceptible individuals in the premises. In every case here, they were applying the same chemical, which is fipronil, which is marketed with the trade name of Regent, for use against a broad range of agricultural pests. And we have the animal version, which is marketed as Allianz. If you look at the upper left image, by applying fipronil directly on the pasture, what is happening is that the largest population of susceptible ticks in the farm that is present as larvae in the pasture is being removed, basically leaving a few resistant individuals that may always be present to be the ones that will climb on the animals and pass their resistant genes onto the next generation of ticks. This is likely the fastest method to speed up resistance, because we're truly removing at once the largest population of susceptible parasites which is in the farm. On the second case, you notice that they are applying Allianz on the back line of this Cebu in that cattle chute. And by doing so, not only are they wasting their money, because this breed is resistant to most external parasites in the tropics, but they're also treating an animal that may allow a few of those susceptible ticks to survive and maintain part of the so-called population in refugia. The fact that they are resistant to ticks does not mean that a few ticks will still be able to infest the animal and manage to successfully complete their cycle. We will see in a minute what is considered as a resistant breed, and it does not necessarily imply that they will repel all the ticks that climb on the animal. So another important principle to a good control of parasites, which is applying target selective treatments to the animals that truly need it, is obviously being ignored here. If you notice on the lower left drawing, selective treatment usually targets the young individuals that are usually the most heavily parasitized and that will benefit the most from the effect of the treatment. And at the same time, it serves to leave some untreated animals in the farm that will maintain certain loads of susceptible parasites, so that the efficacy of the drug is not terminated so quickly. Finally, if we look at the rural farmer on the lower right, what he's doing is he's hand spraying a Regent. It's not a bad method, but I would argue that the technique must ensure that you wet the animal thoroughly, starting by the legs, working its way up all around the animal, and particularly in areas where the ticks tend to concentrate, which are not visible and is usually between the legs. As you can see, this was not being done here, and he was also distributing a 20-liter backpack sprayer among more than 10 animals. In reality, each one of those backpack sprayers is intended for only 4 or 5 animals. So the treatment dose was given is obviously below the recommended one, which is another factor that is going to favor the appearance. This was my final slide. On the next video, I will present some field cases of parasite resistance that we have worked in the last couple of years in this part of Colombia. Two of them will be on the cattle fever tick, which constitutes a real problem to raising cattle in some farms. And another one will be a flock of sheep that had severe losses by the barber pole worm and that had become totally resistant to every dewormer that we have in the country. And again, thank you for watching this presentation, and I look forward to have you in future ones. Bye.